present a little bit of a special problem there, because now we've got two lipid membranes that we've got to bust open in order to get that genome out of that virus. And so that is where we see fusion. So if we have an enveloped virus, fusion follows endocytosis. So what happens here is we have our vesicle inside our cell. So this has our enveloped virus inside it. We have the endosome, so that vesicle is going to fuse with the endosome. What happens when a vesicle fuses with the endosome? Do you know? What happens to the environment inside that vesicle? What did you say? It has to do with pH. What does pH do? It drops, right? It bottoms out, right? So when the vesicle fuses with the endosome, that environment inside that vesicle becomes very, very acidic. And so the pH drops, the environment is very acidic. It's a very hostile environment for that virus. So the virus is kind of on borrowed time at this point. It has got to get out of that vesicle or it's going to be destroyed. How do we know, what do we know about the interactions holding the proteins together that make up that capsid? What type of interactions do we have there? Are they covalent? Nope, they're hydrophobic, right? So we've got hydrophobic interactions, we've got electrostatic interactions, all dependent upon what? Hydrogen bonding, all screwed up by changes in what? pH, right? We screw up the pH, we're going to disrupt the hydrophobic interactions, we're going to disrupt the electrostatic interactions. If those capsid proteins deteriorate, if the bonds in those, or the interactions between those capsid proteins deteriorate, we now have our genome sitting there in this very acidic environment. Is that good for our genome? No. Okay, so our virus is like, oh gosh, I've got to get out of this vesicle. Okay? The way that it does that is that vesicle is going to fuse with the lysosome. What do lysosomes do? What's the lysosome do? Yes, Megan.
Stores things.
Stores things, yep. Okay, so that vesicle is going to fuse with the lysosome, and so we now have this big, huge vesicle. Lysosomes, endosomes in general, these are vesicles that sort of get junk out of our cell that we don't need. They take it, they either trash it or store it, depending on what it is. Okay, but in general, it's going to cause that vesicle to lyse, to open up, and in addition to the membrane, the enveloped membrane, and now I've got my nucleocapsid released into the cell. Nucleocapsid, by the way, is just the capsid in the genome, right? So I've lost my lipid envelope in this process. Okay, so not only have I gotten my virus out of the vesicle, I have now gotten my nucleocapsid out of the envelope through this process. So fusion is the process of that vesicle fusing with the endosome and the lysosome in order to release that nucleocapsid into the environment. All right, questions about that? All right. Next is uncoding. Uncoding is probably the process that we know the least about. It happens primarily simultaneously with penetration. So as soon as the virus particle penetrates the host cell, uncoding happens. So uncoding is the process of releasing the genome into the host cell. So we have, I'm going to go back for 1 second. So if we look at this, we have our nucleocapsid now in the cytoplasm, and now we're going to uncode, which is basically opening up those proteins that make up the capsid and releasing the genome. So uncoding is release of the viral genome into the host cell. So it's disruption of those capsid proteins, which allow for the genome to begin being replicated by that cell. So again, uncoding occurs almost simultaneously with penetration. It occurs right after penetration. They're usually tightly linked. If we really don't understand it very well, we know that there are conformational changes that occur in the capsid proteins that disrupt those hydrophobic interactions, that disrupt those electrostatic interactions, opening up the capsid and allowing for release of the genome. So penetration is really all about getting the entire virus particle inside the cell, and then uncoding is about releasing that viral genome into the cell so that replication can occur. All right. That brings us to genome replication and genome expression. These are the two phases that I'm going to kind of gloss over today because these require a lot of sort of processing and thinking about how different viruses do it. So I want to try and just get through the 8 stages today. So I'm going to come back to these later this week. But what I do want you to know about these right now is that the replication strategy we know is highly dependent upon what the genetic material is. Is it DNA or is it RNA? That is probably the most important thing that determines how the virus replicates. We also know that controlling gene expression, so sort of the hand-in-hand piece with replication, is expression. And we know that controlling gene expression is a major influencer, again, on the course of the infection. Is it going to be an acute rhinovirus, common cold, that we're able to manipulate the expression of those genes, or is it going to be this latent herpes virus infection that we cannot manipulate expression, we cannot control expression, and that virus is going to be able to sort of go dormant and downregulate those proteins and then upregulate them when it needs to. So gene expression is going to play a major role in determining the overall course of a virus. So in 1971, a guy by the name of David Baltimore classified viruses, and he came up with 7 different classes to sort of help explain viral replication depending on the genome, depending on what the material was. And so this is one picture that I think sort of helps us see what's going on with these viruses. So if you can see, we have 7 different classes here, all merging to mRNA, positive sense mRNA. So all of these arrows are getting us to mRNA. And there's another picture, I think it's better. And here's another picture that I like too. So again, this is kind of walking us through all 7 of those classes and getting us to positive sense mRNA. Why do you think that our focus when we talk about viral replication is figuring out what we start with and how we get to mRNA? Why does that make sense?
Could you repeat that?
Why is our focus when we are talking about viral replication only on getting to the mRNA?
Because that's what the host cell interacts with in order to make more copies.
Okay, because that's what the host cell needs in order to make more copies. Once we get to mRNA, positive sense RNA or mRNA, our host cell knows what to do with that, right? It's going to translate that into protein. In every single case of all 7 of these virus strategies, once we get it to positive sense RNA or the mRNA, it's going to be translated into protein, okay? And then we're going to make more virus particles. Now, how we get there differs depending on what we start with. Okay, so we'll talk more about that next time. So class 1, these are our double-stranded DNA viruses. So we know our cell knows what to do with that. It's immediately going to be transcribed into positive sense mRNA. Okay, so that's class 1. They're one of the easiest ones. Our cells know what to do with it. We're going to transcribe it into positive sense mRNA. Class 2 are our single-stranded DNA viruses. So these are our class 2 viruses up here, single-stranded DNA viruses. So they have to go through an extra step to copy themselves, to replicate themselves and make a complementary strand to become double-stranded, and then they can be transcribed into mRNA. Right, so that's our class 2 single-stranded virus. Class 3 are double-stranded RNA viruses. So that's these guys. So our double-stranded RNA viruses have both positive sense RNA and negative sense RNA, right? So they have a strand that looks exactly like the mRNA, and then they have the complement of that strand. But we don't really care about that complement strand. We've got the positive sense strand in here, right, in this double-stranded RNA. We can use that positive sense strand to translate into our proteins. Okay, so that's class 3, our double-stranded RNA. Class 4 are our positive sense RNA molecules. Now, class 4 is kind of interesting. So if we look at class 4, they start with positive sense RNA, then they go to negative sense RNA, and then they go to positive sense again. Think about that one for a second. Why in the world would our positive sense RNA viruses go to negative sense and then go to positive sense?
Is it so that they can create the template?
So they can make the template, right? So they can make more of themselves. What are they using this negative strand for? They're using it as the template to make more positive sense. So the negative sense is simply serving as the template so that they can make a complement strand. And a complement strand of the negative sense is what? The positive sense. Okay, so they're using that negative sense strand as a template. All right, class 5, those are our negative RNA viruses. So again, we know we just make the complement of our negative sense strand, we get our positive sense strand. Class 6, these are our retroviruses. Class 6 here, sorry. So these are our positive sense RNA viruses. We know they go to DNA. So they remember, if you think back to the video that we watched on HIV, so we use this positive sense strand as a template to make a single-stranded DNA molecule, and then we use that to make our template to make our double-stranded DNA molecule. And now we're back here to this, which we can then go straight to mRNA. Okay, so those are the retroviruses. Those are class 6. In class 7, those are our, we almost never see viruses of class 7. Class 7 are double-stranded DNA viruses with RNA intermediates. So that one's not even on the chart, so you don't have to worry so much about that one. So don't worry about class 7. We're just going to focus on the first class, the first 6 classes. So what you will be responsible for here is knowing what the virus starts with. And again, I'm not going to ask you to memorize the classes. I would tell you, if I have a negative sense RNA single-stranded virus, how do I get to mRNA? What do I need to do? What steps or what strategies do I need to go through to get to mRNA? Once we get to mRNA, our cells know what to do with that. Now, there are some slides through here. I'm going to skip this, but I will give you a handout. I'll post a handout with all of this information on it. Alright, so once we get through gene replication, expression, again, I'm not going to spend a lot of time on gene expression because this is very similar. Viruses carry out gene expression in almost the same way that host cells carry out gene expression. So a lot of these things you are familiar with. So control of viral replication is going to be achieved through replication of gene expression. The methods are going to vary, but a lot of these things are the same as how host cells use it. So we talked about those segmented genomes before and how those can be advantageous to viruses. Remember in the video that we watched on HIV, it talked about the virus-encoded protease, which was responsible for sort of chopping the big viral proteins into smaller molecules. So that's another way that viruses regulate gene expression. We have all of those same sort of signals that we talk about in host cells, the ribosome binding sites, the poly-A tails, splicing of splicing of RNA to create different genes. Again, we see that in host cells as well. We know that specifically DNA viruses, again, use transcriptional regulators, just like host cells do, to simulate polymerases. A lot of these things, again, I'm not going to spend too much time on this, but know that viruses utilize many of the same expression and regulation strategies that your host cells use in order to regulate their own gene expression. So you typically see, again, this is another example of virus manipulating the host cells to do what they want them to do. All right, so I just have a few minutes left. I want to move on to assembly. So this is our next phase of replication. This is basically where we are collecting all of the components that we need to produce that infectious virus particle. And different viruses do it differently. But the idea is we are replicating our genome, we are producing or translating that genome into the capsid proteins that we need, and at some point we have to start putting those things together into an infectious virus particle. Now, what that point is a hard target to pinpoint, and it varies from virus to virus. Basically, what virologists think is that at some point during that replication time period, the components, the genome and the capsid protein, reaches this sort of critical concentration inside the cell, and the virus says, okay, we need to start putting together, we need to start assembling the virus particles and sending them out to infect other host cells. So we've reached this critical level, we have enough genome, we've got enough capsid proteins inside the cell, we've reached a critical concentration, and it's time to start putting them together and sending them out. So this is where we form the basic structure of that virus, and again, different viruses do this in different places of the cell. So viruses that fall into the coronavirus, poxvirus, or reovirus families have their collection points in the cytoplasm. Adenovirus, parvovirus have their collection points in the nucleus because they're DNA viruses. And retroviruses do it just under the inner surface of that membrane because they're going to bud out of that membrane. Some viruses use what we call inclusion bodies. So these are just little pockets inside the cell, little membrane-bound pockets where they sort of store all of their components until it is time to start assembling them. Okay, so we just had Valentine's Day, so let's think of this analogy, right? Let's say that we're making Valentines for our friends, and we're gonna have a big purple heart, and then a medium red heart, and then a small pink heart. And we're gonna cut out all these hearts, and we're gonna tape them all together. We're gonna write something nice about our friend on that Valentine's Day program. Okay. So the way that we might go about this is we might cut out all of our purple hearts, and then cut out all of our red hearts, and then cut out all of our pink hearts. Okay, so we've got all of our components made, but we haven't actually put anything together yet to make a Valentine, all right? But we keep getting interrupted during this process, right? So we have to put our hearts someplace safe where our little brother will not tear up and destroy our hearts while we are waiting to make our Valentines. So we have our desk drawer or whatever where we store our components. So that is sort of like the inclusion body inside the cell. This is the vesicle that a virus uses to store all of its components so the host cell won't destroy them before we can get them assembled and put together. All right. So now let's say we have 20 friends in our class. We've made 20 of all of our different colored parts. And so we have reached that critical concentration where we are now ready to start putting together our Valentines and putting our components together to make a fully functional Valentine. Right? That's exactly what the virus does. It stores up all of these components until it has reached a critical concentration and it's ready to start putting them together to make a fully functional infectious virus particle. There might be something wrong with my analogy that I'm comparing Valentines to infectious virus particles, but I don't know, it just came to mind. I don't know what a psychologist would say without saying that. Okay. Hopefully that illustrates the point. Alright, so assembly is driven by lots of different things. Again, we have these inclusion bodies. It can be a simple interaction between the subunits of the capsid, so as soon as those protein particles come together, we're ready to start assembling. It can be much more complex and involve like scaffolding proteins that are virus encoded and cellular scaffolding proteins. It really just depends on the virus of how they go about the process of assembly. All right, the next stage is maturation. This is where the virus actually becomes infectious. Okay, so this is where the virus actually becomes infectious. We don't know a lot. Again, maturation is one of those phases that is sort of poorly understood, that we don't know a lot about it. Some viruses, as soon as they assemble, they are mature. Some viruses, like HIV, for example, has to undergo another step or another process in order to become a mature infectious particle. Oftentimes, there are structural changes that happen within the nucleoproteins, so within the proteins that are involved or attached to the genome. There are proteases or other cellular enzymes that are involved and that contribute to these conformational changes that allow that particle to become mature. So sometimes maturation happens within the host cell before it is released, and other time maturation happens in the extracellular environment, so after the particle is released. For example, the HIV video we watched, you see that HIV becomes infectious after it leaves the cell. All right, so maturation and exiting the cell can sometimes flip-flop depending on the virus. So sometimes maturation happens inside, sometimes maturation happens once the virus has been released from the host. All right, speaking of release, that is the last phase. So we talked a little bit about this already. We know we have lytic viruses that exit a cell via lysis. So these are your non-enveloped viruses. So the cell lyses, cell bursts open, and that is the way that the virus exits that cell. The other way that we know viruses exit a cell is through budding, which is basically exocytosis. And they leave, when they leave, they get that membrane coating or that lipid envelope. There are advantages and disadvantages to both of these types of exits, which we will talk about when we talk about viral host interaction. But again, there is a balance that all viruses have to maintain between making more virus and infecting more host cells, yet keeping their host alive and not destroying so many cells that the host dies. Okay, so that brings us kind of to the Ebola case, which we are going to talk about on Thursday. So Ebola is a horrendous virus that causes tremendous death and destruction in its hosts. But in terms of if we were to evaluate it, in terms of how good a virus is it really, it's probably not the best, not the most efficient virus that we know of, because it very rapidly kills its host, right? So Ebola can spread with tremendous strength, right? It has great ability to replicate and exit a host cell, but every time it exits a host cell, it kills the host cell, and you lyse it. And so it replicates so well and so fast and kills so many host cells along the way that the host itself is usually not able to survive that process. So this case study that I just gave you is we're gonna walk through sort of how Ebola goes through these replication strategies. So this is a way that we're gonna sort of look at an example of all of these different phases of replication, and particularly how Ebola does it. Any questions? All right, so you should come prepared on Thursday. You should read this before class. You should answer the questions in here. Again, it's not something I'm going to take up or you're going to turn in, but you will be expected to participate in a discussion of the questions. I may call on you to answer any question at any given time after you have had a chance to discuss it with your groups. So come prepared to answer the questions on Thursday. It's supposed to snow on Wednesday.
Yeah.
It was supposed to snow on Saturday too, though.
Really?
Let's hope,
I didn't hear that.
It was.
let's hope our Wednesday snow
Oh, yeah.
is a lot like
It
our
was either
Saturday
or.
snow. That's right.